So really quickly, before we start, well, we have started. Let's take a look at some of the submissions. So here's the way this is going to work for the rest of the term. When I wrote up there on the first page, let's see, don't add your name to the assignment or the file name. I do that because I'd like to open up some of your submissions in class, and I want to make sure that everyone's comfortable with this. So it's anonymous. So I'm not going to be, if your name is up there, I'm not going to open your file today. If you want in-class review of your work, don't name or sign or put your student ID on your documents. Okay. So here's the first one that came in. How many steps and rearrangements to go from left to right? Does this make sense? Take a minute, half a minute maybe. See if it looks like this works out the way that the author intended. What do you think? Who says, I agree that's a reasonable answer? Does anybody have any objections to anything that's happened here? Please do.
Can the AA go together and then invert, or does it have to be like that?
So, is there what's another way to do this?
To duplicate, yeah.
So let's see. If we were going to do this, did you submit one of these?
I submitted it already.
It's going to be hard to find yours though, because I didn't ask anybody to put their names on it.
Yes, I
so
didn't put my name on it.
we're still working through this process of how to annotate things and make them useful. So did the A and the B have to duplicate together, or could they have independently duplicated?
Yes.
So what's the issue with independent duplications in this case?
Takes more steps.
Say it louder.
Takes more steps.
It takes more steps. So you could absolutely have a duplication of A and then a duplication of B to make that extra copy of B and A over there on the right side. But that's two steps when duplicating A and B together could be a single step as it's drawn up here. Sir?
I have a question. First question is, could you have had the duplication first and then the deletion? The second follow-up question to that is, you might have mentioned this earlier, when we have a deletion, if the two bases or nucleotides are next to each other, can the deletion happen and delete both of those nucleotides, or each deletion have to delete one nucleotide at a time?
So two questions. The first question I'm going to answer with, I'll restate the question so everybody hears it. The first question is this necessarily the order of steps? Does somebody else want to answer that? Has anybody figured this out yet? Does the deletion have to come first and then a duplication and then an inversion? I see a head shaking. That's a good sign. So everybody should think about, so I'm not going to answer your question, but do think about this. Do these things have to happen in this order? So what you should maybe do is play with this after class if you've got time. Try doing this in a different order and see if you get the same result, outcome. Now your second question was, it was similar to a question that was asked last class, can multiple things happen at the same time, essentially, which is absolutely yes. So you could have a deletion and an inversion happen at the same time. It's just not very likely. And please ask a follow-up if you want. The other point that I want to make here is that these deletions are not necessarily single nucleotide deletions. So C, what I've highlighted here is locus C or area C, could be 100 kilobase pairs, 100,000 base pairs of DNA sequence. So it's not just, you asked specifically about one nucleotide or two nucleotides. I'm just trying to make the point, which is ancillary to your question, that we're talking about usually much bigger pieces of DNA moving around than just a single base pair or two. So follow-up question? Please.
I guess the follow-up question is, if you have the deletion on, like, you're deleting A, B, and D, E, so you can have those in one step?
Sure. So in blue,
Okay.
could you have that deletion delta, the triangle for deletion. Could those happen at the same time? Sure. Perfect answer for class. This is, again, no right answer. There are lots of ways, and that's one of the reasons I wanted to get your submissions from that while you're waiting exercise, so that I could show you, and I will at some point, how many different answers all of you probably came up with to go from the structure of the chromosome on the left to the structure on the right. The second page to this, which some of you send in answers to. This submission really highlights an important point. Sir?
For the duplication, can you add any more, or does that have to be on top of the bottom?
In the previous example.
Right.
Can the duplication happen?
Anywhere, like, could you just stick in A and B in the middle?
Yes. So you could copy A and B and insert them anywhere you wanted on the chromosome. But why in that example we were just looking at did the author of that solution put A, B together next to the original A and B? So the outcome was something like A, B, E, or something like that. We started with a single A and a single B, and there was probably a C and a D and an E down here. Something like that.
Right.
Seriously? Thank you very much. Too many pieces of technology up here. So this was approximately the starting situation. Maybe the letters are different, but it'll illustrate the same point. If you want to make a duplication of A and B, there's one copy on the left side, there's two copies on the right, there has to be a duplication. Where do you want to stick that extra copy of A and B? Where do you want to insert it on the chromosome on the left? Somebody tell me. Between which two letters over here do you want to stick that piece of DNA? Between C and B, C and D? Doesn't matter. You could put it in there between B and C. That's fine. What's the benefit of doing it that way? You're already at the situation that you're trying to achieve on the right. What happens if in red you stuck A and B between C and D? Then you have to have two separate deletions to delete C, so the intermediate state would look like this. A, B, C, A, B, D, E. So now what do you have to do to get to that last chromosome? You have to delete C, and you have to delete D. So now we've got 3 steps. We've got an insertion, we've got 2 deletions. So where you put that duplication of A and B ultimately affects how many total steps that you have to achieve the goal, the order of loci on the right.
You said that you have a total of 3 steps, and you can get 2 steps of the deletion, but you just said earlier that you could have 2 deletions, I don't think so.
That's right. So again, bottom line, let's highlight the important key messages of these exercises for everybody. One is that in reality, in nature, who really knows what happens for any specific instance of chromosomal change? It could have happened like happened in red here. It's possible, it's just not very probable. The more steps you invoke, the less likely it is to have happened that way. Doesn't mean it didn't happen that way, it just means it's less likely. So for the purpose of life, for the purpose of how things really work in nature, and probably for you the purpose of exams, any of these answers would be correct. But I do want you all to take away the idea that more steps is not as likely. Write it down, underline it. It's possible, just less likely. Okay. also some questions about fissions, fusions, and translocations, so I thought it might be useful to do a side-by-side comparison. So let's do a fusion on the left. What is it going to look like? So I want to point out, which it says down here at the bottom, which you can't see, now I'm drawing interphase chromosomes. So a single line, vertical line up here represents what, do you think? A chromosome, which is the same thing as a double helix, right? So we've just zoomed way out. One double helix, one chromosome, one vertical black line. Okay, what do you think the white circle is? Centromere. Okay, so we've got, on the left side here, we've got two pieces of DNA, two molecules of DNA, two chromosomes. What are they going to look like after they fuse? Let's say they fuse together in that orientation that's shown. So after a fusion, there's going to be that chromosome with its centromere. Then what do I do next? Sure. Right. So you'd have the black and blue chromosomes just tail to tail, telomere to telomere, fused, stuck together. And there would also still be a centromere up there. So now you have a problem. You've got a dicentric chromosome. Chromosomes don't like having two centromeres. We'll see why when we get to DNA replication, but in the meantime, two centromeres is bad. So that's a fusion, chromosomes sticking together, usually telomere to telomere. So you just add the two double helices together.
So for the purpose of this, though, you're assuming that this is like acrocentric, not telocentric, right? The bottom ones?
Sure.
Because
these are acros.
telomere to telomere, not.
Right. These are drawn, so regardless of the shape of the chromosome, any fusion that's end to end will necessarily be telomere to telomere.
Even if it is centromere, technically?
Because every chromosome, even the telocentric chromosomes, have telomeres on both ends. So all linear chromosomes on both ends of the chromosome, there's telomere sequence. So if there's an end-to-end fusion of any two chromosomes, it will necessarily be telomere from the end of chromosome 1 to the telomere of the end of chromosome 2. In the second example here in the middle, let's say the black chromosome undergoes fission. Somebody suggest a place where we can fission that chromosome. Use genetic terminology. Where in this big chromosome should I break it? Yeah, guess something. Say something. Anybody. It's Friday. Everybody should be relaxed and under the centromere. So what arm? This is what shape of chromosome? What centricity? Metacentric, submetacentric, acrocentric, telocentric? Submetacentric, I heard from most of you. So this is submetacentric. So if I want to break underneath the centromere in this drawing, which arm of the chromosome is that? Is that the P arm or the Q arm? Q, the long arm. So we've got the Q arm. So let's say I break that chromosome right there, fission. What's the result? Two chromosomes, two separate molecules of DNA. So we call them two chromosomes, but what?
Chromosome would be two helixes.
So we've got a chromosome on the top that has a centromere, and something that we're calling a chromosome that's the little tiny fragment of the Q arm that has a problem. No centromere. Which, again, we'll learn later is bad. You might already know why. It's because it can't segregate faithfully during mitosis. So when this chromosome, without the centromere, replicates, it'll replicate, probably. There'll be two copies of it at some point. But when it comes time to divide the cell, There's nothing for the microtubules to grab onto. There's no centromere to make sure that one copy of that little fragment of DNA goes into each daughter cell. So 2 centromeres, bad. No centromeres, bad. 1 would be perfect. So the most important point, I think, and the one that I think most of you who said, let's work on this again, were really wondering about was the translocation. I heard a lot of how does fusion and translocation differ? So we've already seen an example of a fusion, end-to-end connection of chromosomes. What happens if we have a translocation between these 2 chromosomes? Let's say they translocate at their centromeres, just for the sake of moving ahead. So what that essentially means is that both of these chromosomes break wherever we're talking about. In this case, I said we're going to choose both centromeres. So both of those chromosomes are going to break. And they swap bits. It's a very technical term. So that what you wind up getting on top is a chromosome that has some black chromosome there, and the bottom half is blue, and one where the top half is blue, and the bottom half is black. Yes?
When they say translocation, should we always assume that it's a reciprocal translocation?
So there are lots of different types of translocations. What I just drew was a reciprocal translocation. I love your questions, but I always feel bad when I answer them because I'm always saying, well, you know, this is the answer for now, or this is my cop-out answer. I want to, since you raised this point, I want to make a really important point about right answers for this class. If you ever get to a point in class or on an exam where you think, is this a trick question? One right answer, multiple right answers, how am I going to prove to Dr. Ross I know what I'm talking about? All you have to do is write your answer and justify it. That shows me your understanding. So if you want to say, I drew it this way because I assumed it was a reciprocal translocation, or I assumed you were asking about a reciprocal translocation, that's how I'll grade it. So you tell me what your assumptions are. That helps me know if you understand the material. I grade your answer based on all of the assumptions you've told me you've made. So I don't want to say always assume that it's reciprocal translocation, because sometimes it's not. But if you feel like at some point ever you need to make an assumption, just tell me and we'll be good. Anything wrong with these last 2 chromosomes after this reciprocal translocation? Has anything really changed?
Shouldn't the P arm one be longer?
Oh, well, let's see. If you're going to be nitpicky, let's see. What's the P arm, the bottom one? Yeah, maybe. I'm not going to edit it. But yes, so you could definitely, you want to make sure, what's the source or what's the key concept of your question? Why do you ask?
I just want to make sure, like, if we are doing a translocation, like he was saying, it is reciprocal, or if not, then is the DNA just lower than it was in the fission?
Right. So I think the core concept here is, with a translocation, you don't lose or gain DNA. So you're asking me to make sure that I've drawn all of the total lengths of the chromosomes exactly the same in the before and the after. Because all that's happening is chromosomes are breaking and switching parts.
And the translocation is also kind of just an insertion, right? In the sense that you're just taking it from one piece of DNA for sure and then moving it to another.
Right. So translocation is like cut and paste. If we want to use since we're in a tablet class, let's use a computer analogy. You cut part of chromosome out of one, you paste it into another one. There's no change in DNA content. Okay, I think if I remember correctly, this is the last review / we need to clarify this. chromosomes, chromatin. So we're slowly getting there, which is good. So I decided to show you from, I think this was day 1 or day 2, one of our first assignments. Pick a photograph of a chromosome, a micrograph here, and then draw how that's related to a double helix. Right, so this is one of your submissions. Thank you very much. This is one of the really, really nice ones that illustrates the point that's necessary to make. You start with the double helix. What's this made up of? What are all of the components that you could tell me right now that make up a double helix of DNA? Sugar, nitrogenous base, phosphate, atoms, small organic molecules that all get connected together to make the double helix. What happens when we go from that step to this step? What are we adding into DNA? Proteins, the histones. Together, DNA plus protein makes the thing that we call chromatin. So that's chromosome versus chromatin. Chromatin is the stuff that makes up what we look at and see, oh, that's a chromosome. So technically, a chromosome is just the double helix of DNA without protein, technically. You add in some protein and you package it in a way we can see it. That stuff, the DNA plus protein, is chromatin. So chromatin is more of an ingredient. And the chromosome is like the final product. In fact, because I love analogies so much, let's try this. Let's hope the internet holds up. Hey, that'll work. So, let's see. Check out those 2 rotinis. They're like 2 chromosomes. They're not double helices. They're single helices, but play with me. So we've got rotini, a type of noodle. That's my analogy to the chromosome. It's a finished product. It's a thing. We observe it. We see it. There's an object. It's a double helix. It's a rotini noodle. What's the rotini made out of? What are the ingredients that make up a rotini? Italians, come on. Flour, water, egg. Right? So we've got chromatin. So the concept of chromatin versus chromosome. Chromatin is the stuff that makes up a chromosome. Flour, water, and egg is the stuff that makes up a rotini. So chromatin is the stuff that makes up the chromosome that we see. It's the ingredients that produce the final product. Chromatin, DNA plus protein, add them together, you get chromosome. So chromatin is a thing, it's ingredients. It's the raw material from which chromosomes are made. DNA plus protein. Okay. So a bit of practice with interpreting fluorescence in situ hybridization experiments. There's a series of these that those of you that have downloaded the slides can see. There's 2 or 3 of these. So I thought this might be useful to you, I hope, because we're going to look at the same metaphase chromosome spread. And we're going to look at different patterns of fish signals, the blue drawings up there. And we're going to figure out what's different between the different scenarios. We're going to use the same chromosomes in each of these examples. We're just going to change where the fish probes are located, hybridized. And that's going to hopefully tell us something useful about how we use cytogenetics to learn about chromosome structure. So here's example 1. We've got 4 pairs of metaphase chromosomes. So you might also think about these as looking like this. I've just drawn them in a different way so that the chromosomes aren't crossing each other. They're next to each other, side by side over here. So we've got 4 pairs of double helices, 4 pairs of chromosomes. What is the location of the blue fish signals tell us about, what can we infer? What do we know now about the organism that this chromosome spread, this nucleus came from?
And that the two leftmost chromosomes are paired like
Okay, so how many, yes, what's the ploidy of this individual? Haploid, diploid, tetraploid, does it have one copy or two copies or four copies? This is diploid. Because when we look at metaphase, this is an important but subtle point. When we look at metaphase, that's after DNA synthesis. Which means that back in interphase, where we measure ploidy, there were two copies. There were two double helices. After synthesis, there are four. 1, 2, 3, 4. So after, in a diploid individual, after synthesis, you have 4 copies of each chromosome. It's a diploid individual. And then the same individual back here gave an excellent answer to the second question. What do the locations of these fish probes tell us about this organism, or its chromosomes at least?
Their relationship to each other?
Their relationship. So we've got 4 chromosomes. If I didn't have those blue fish signals on there, how would you know which 2 pairs of chromosomes are supposed to be paired up? This could be chromosome 1 and that could be chromosome 1. And this could be chromosome 2 and that could be chromosome 2, right? If we don't, because all the chromosomes look the same. They're the same size, they're the same shape. Without this information that the fluorescence in situ hybridization experiment gives us, we don't know which of these objects are identical. They're paired up together.
Dr. Rob? This might be kind of a side question, but since we're talking about today. When you were talking about translocation, and you're talking about the replication, during meiosis, when my chromosomes do the crossing over, is that considered a translocation?
So yes, a side question, I'll answer it briefly. During meiosis, when crossing over happens, does that count as a translocation? Technically, yes, but that's not what we're talking about here. But yes, that is chromosomes breaking and exchanging parts.
Okay.
Yep. Okay. So I'm not going to talk you through this one. Spend a minute by yourselves, individually, thinking about answering the same 2 questions for this example. Okay, if you haven't already, talk to a neighbor, convince them that your answer is correct. Unless you already have. That's the most annoying thing I've ever seen.
Isn't that
at least
annoying?
today.
Oh my god, I have to like draw and then like erase and then it shows up. Yeah. I don't get it.
Okay, so what's the consensus? What are you agreeing as answers? So I've heard tetraploid. Does anybody have another answer? This might be one of those ones where there's more than one right answer. Anybody else say something other than tetraploid?
It could be diploid, it's just that an insertion happened where they each got a set of that same.
So now we're talking about thinking creatively. I would have said tetraploid. One possible right answer. Absolutely. Could this be a duplication of the part of the chromosome that contains the sequence that this fish probe hybridizes to? So this could be tetraploid. Or you might have had that part of that chromosome duplicate onto the ends of those two chromosomes. So this might be chromosome pair 1 and chromosome pair 1, and that could be 2 and 2. And that might explain the results you're getting. Oh man, so many answers. I favor tetraploid, but that's just because that's what I was thinking when I made this example, but you're absolutely right. That's another entirely reasonable argument for why you would see this pattern. So if that part of the chromosome that's blue happened to have duplicated, you might see this pattern, even though you're diploid. Sir.
If we were looking at this as a karyotype, when you're marking as 1, should that just be the one single strand? Or why do you have 2 ones for that? Because then when you do the karyotype, you have two lines next to each other.
So let's assume that this is a great question. Getting back to that same underlying thing about double helices, molecules of DNA, chromosomes. When you look at a karyogram, if you look at many of the pictures of human karyograms we've looked at, for example, up here in blue in the upper right is what we normally see on a karyogram. Sometimes, though, in a couple of lectures, I've shown some pictures from Google Images search for human karyogram, where the chromosomes kind of look like that. So they're not crossing each other, they're laying side by side. This is equivalent to that. So you've got 4 molecules of DNA, 4 double helices. The same is true there. So you've got 2 copies, you've got 2 pairs of copies of chromosome 1 here. You have, in the karyogram, 2 pairs of copies of human chromosome 1. And the same would be true if they looked like these ones. That's 4 double helices. It's 4 copies of the chromosome, the same as is drawn here. Given the discussion we had on the last question, I'm just going to move ahead with the answer to this one. So what's different between the previous example and this one in terms of where the fish probes are located?
It's on the top part of the Q arm versus the bottom part of the Q arm.
Right. So there's 2 different locations. Here, the fish probes are at the bottom of one pair of chromosomes. Here, they're at the centromere, near the centromere of the long arm. Is this, think back to the last slide, could have been tetraploidy, or it could have been diploidy with duplication, the answers to the last slide. Is that also true for this case?
It could be diploid with inversion.
It could be diploid with,
Or would the fish probe not pick it up because the sequence would be backwards?
could this be tetraploid?
Oh, that's what I was thinking. I was thinking tetraploid.
So if this individual is tetraploid, why aren't the fish signals all at the same place on 4 pairs of chromosomes? Because there was an inversion.
But the question is, would it read the sequence if it's backwards? Does the fish probe not care?
That's correct. In fish experiments, It doesn't matter what orientation or anything the chromosome segments wind up in. We'll still see it. So it could be diploid with a duplication, or it could be tetraploid and there's an inversion.
I thought of those two scenarios with the diploid, duplication, and tetraploid.
Yep.
But you said that the easiest answer is always the correct one. Then wouldn't it be easier just to say that it's diploid?
Oh, now we're making judgment calls about which is better, diploids or tetraploids. I'm sorry, I'm kidding. I'm poking fun, but hopefully in a nice way. So is it better, is it more likely if you don't know the organism you're looking at, is it more likely diploid or tetraploid? If I just handed you a microscope slide and said, here's a metaphase spread of some species. I honestly don't know the answer. I mean, we'd have to go to Wikipedia probably. That's what I would do. I would go to Wikipedia and find out how many diploid species are there on the planet versus how many tetraploid species are on the planet. Well, plants tend to be more tetraploid than, and so there's a higher frequency of tetraploidy in plants than there are in animals. But you still see some tetraploidy in animals.
Especially like cellular.
A lot of salmonids are tetraploid, for example. There are a lot of salmon and trout species that have 4 copies of every chromosome. So fish, for some reason, not fish the technique, but fish the thing that swims around, for some reason tend to be more able to accommodate having multiple copies of their chromosomes. So in short, we shouldn't use ploidy necessarily as a metric for determining which of these things is more likely. But this is a very valid question. Okay. Last one, just so I've recorded the answer to it. What happened here? There might have been a deletion. So what would explain seeing just one pair of fish signals? Could be that, or could be haploid. Oops. So if you're an organism that only has 1 copy of every chromosome, this is what you would see if you did fluorescence in situ hybridizations. So I hope I'm making you comfortable yet uncomfortable with the idea that there can be multiple explanations in science, because that's the truth. When I do these cytogenetics experiments in my lab, I have to consider all of these possibilities when I look at chromosomes. Then what would be a really good exam question would be, what would you do next to distinguish between these 2 possibilities? What would be the next experiment? What would be the next piece of data you would want to have that would help you rule out one of those? To be continued. So if this pattern is due to a translocation, you still need to explain why we don't see that second pair of fish signals.
Well, it could be translocated off the...
So what would happen if there had been a translocation where that chromosome used to have those blue signals, it broke there, what might have happened? So this is basically looking like a deletion. It's breakage and then loss. Well, if these pieces move on to another chromosome, say there, then that's where we would see.
Unless it moved on to the one that has the fish probe.
Oh dear. So, in red, what happens if there's a translocation of those there? What do we expect?
A duplication.
It looks like a duplication. What do we see when we look in the microscope? We see longer, bigger stretches of blue fluorescence on that chromosome. So that chromosome looks a little bit longer compared to the one that had the translocation. It's got more blue fluorescence because it's got twice as many copies of this DNA sequence.
But you would notice it if it was the only fish probe.
Right, so
Signal.
now in reality, we're not sure if we would actually recognize that there would have been, that this had actually happened. If you look in a microscope, you say, "Oh, is that twice as blue as the last one I looked at?" Doesn't sound very scientific. So this is, again, totally valid hypothesis for what might have happened in this image. So I really, truly, honestly mean be creative when you come up with answers for things like this, because all of these are valid. Quick one.
Couldn't you sequence the DNA to find out you've seen the same chunk of DNA replicated 2 times, and then you would find out it's 2 times?
So bring that question, so could you sequence this chromosome and find out what had happened? Absolutely. Bring that question back next class when we actually talk about DNA sequencing. So we're coming up on that really quickly. But when I'm asking about, what would I do next if I was faced with all of these possibilities, how would I know what really happened? DNA sequencing is the go-to. It's expensive, though. I don't have $1,000 to shell out every time I want to sequence a chromosome, but please do. Go ahead.
Could that be due to a translocation or no? A double end with translocation?
So if there's a translocation, okay. So the goal for today, I'm so glad that we're not going to get to this because I spent some time preparing it, but like I've said, I'd rather answer questions than go through slides I've come up with. So these were my goals for you for chapter 18. This one section that we read in the textbook has a lot of mechanism in it. You saw reactive oxygen species cause guanine to become 8-oxo something guanine, and it only base pairs with A. I'm going to skip this for a second. Oh, by the way, we're talking about reactive oxygen species in this chapter on mutation. These are byproducts that our bodies produce while our electron transport chains, our mitochondria, make ATP, the energy that we use to do everything. So our mitochondria normally produce these peroxides, reactive oxygen species, as we go. And that's presumably why we can go buy these creams that have antioxidants in them, just to neutralize the free radicals and make us look beautiful or something like this. And of course, why you're supposed to eat the rainbow of fruits and vegetables so that you make sure you're eating lots of kale and other nasty tasting things that taste nasty in part because they've got some antioxidants in them. And so this is what, as your textbook explains, happens. You have guanine, and after it's exposed to reactive oxygen species, it oxidizes here, so there's a change to guanine. And this molecule, 8-oxoguanine, now base pairs with A. G is not supposed to pair with A, people. You know that. But our bodies produce the free radicals that can cause this to happen to our DNA. So what I'd like you to do for next time, one of the things I'd like you to do for next time, again, this is not to turn in, bring back to class so that you're prepared to discuss if you've got questions. Here's a short little double helix of DNA that's been exposed to reactive oxygen species. So if it is exposed to reactive oxygen species, what will happen after this double helix replicates? So bring back the sequence of the 2 chromosomes that are produced after this molecule replicates. We'll discuss then. So please turn to Socrative. There's a short, as usual, little exit activity. And while you're doing that, I'm going to put up the list of things to do for next time. 1 or 2 videos to watch, not ones that I produced, but other videos. The answer to that activity that we just talked about on the previous slide, and a reading assignment. Thank you very much for your feedback on Socrative. And when you're done and you've looked at what I'm going to put up on the next slide, you're certainly free to go. Have a fabulous weekend. Stay ash-free. Yeah, good luck.